and welcome everybody. We're just going to wait a minute or two for a few more people to trickle in, and then we'll get started. Well, I guess we've officially cleared 100 people, so let's get going.
Let's go.
Welcome to today's webinar on AI and drug discovery and longevity. We're just going to start off with a few housekeeping items. So everyone who's attending, your lines are muted. But if you do have a question, please enter it in the Q&A box at the bottom of your screen. And in the second half of this hour, we're going to be taking some of those questions from the audience and discussing them with the panelists, and we will be recording. The topic for today is AI for longevity drug discovery, breakthroughs and challenges. Really looking forward to the conversation. I'm your moderator, Kristen Fortney, PhD in aging biology and an AFAR grantee back in the day, and currently the CEO and co-founder of BioAge. A bit more about our host. So AFAR, if you work in aging biology, you're probably familiar with AFAR. They support a lot of talent and a lot of research in the field, deploying nearly 200 million to 4,000 plus investigators, and are really important in fostering young talent as well. I'm going to briefly introduce our panelists, and then we'll go into a longer self-introduction in just a couple of minutes. But we have Ben Blue, PhD, CTO and co-founder of Ora Biomedical, joining us today. Hanadie Yousef, CEO and co-founder of Juvena Therapeutics. Everyone is also a scientist in aging biology, so scientist and founder. We've got all that in common. And of course, Zhavoronkov, PhD, CEO and founder of Insilico Medicine. So yeah, really looking forward to, let's get started. All right, so everyone's seen your names, but let's talk a little bit more about what you do, right? So let's do some brief self-intros, 1 to 2 minutes tops, but maybe introduce, like, what does your company do? And where are you in your drug development journey? You know, we've got the whole spectrum here from like new company to, you know, deep clinical pipeline. And also relevant to today's topic, how are you using AI in your company? What are some of the key technologies that you rely on? Hanadie, you want to kick us off?
Sure. My pleasure, Kristen. Really excited to be here today with everyone virtually. So at Juvena Therapeutics, we are developing biologics for chronic and rare diseases, including age-related diseases. And we're doing it through a unique platform that's AI-enabled that enables us to mine proteins secreted by stem cells in order to map their therapeutic potential to tissues and cells in which they can really have rejuvenating benefits. Once we do these mappings, we fully validate them and then translate them into engineered biologics for very specific disease indications with paths through regulatory to ultimately get them approved by the FDA and into the clinic and to the people who need them the most. And in terms of the different technologies we're really using a variety of both computational, bioinformatic, and AI techniques combined with different data modalities, including quantitative proteomics, single-cell transcriptomics, and other types of multi-omics, high-content imaging, and different really like AI and machine learning techniques such as neural networks, generative AI, and others to really innovate at every step of the process of in silico, in vitro, in vivo screening, and then lead optimization and protein engineering. And so that has today enabled Juvena actually over the last 6.5 years to really build a biologics pipeline with now 2 preclinical stage assets, and actually 1 asset that is we're in the midst of regulatory filing. And we're actually going to be in phase 1 starting early this fall, and it's for a muscular dystrophy known as myotonic dystrophy type 1. It is a biologic that can act across a variety of muscle wasting diseases, and of course, happy to share more a little later on about it.
That's great. Yeah. And Ben?
Yeah, so I'm CTO of Ora Biomedical. Our platform is based on using robotics and neural networks to do high throughput drug discovery with lifespan and aging biomarkers as the starting phenotype. So we start in whole animals, the canonical and lovely invertebrate C. elegans, to look first and foremost at what is modulating overall lifespan to identify gerotherapeutics. from there, our goal is to really expand outward and not just stay under the lamppost of, say, mTOR inhibitors and things that we know work, and actually explore untouched regions of chemical space to find the next generation of longevity therapeutics. So I'm really excited to be on the panel today. Honestly, I think Ora is the youngest of our 4 companies, and really, I just want to highlight the work that you 3 have done translating, good geroscience into actual pharmaceutical pipelines. It's really, really inspiring to be a part of this today. So thank you for the 3 of your companies and your work getting this going.
Yeah, thanks, Ben. And Alex?
So great pleasure to be with you today. These are the companies that are, I would say, maybe some of the most credible in the field. Insilico has been around for now more than 10 years. We were founded in 2014, and now it's our 10th year in existence. We started our own drug discovery journey where we decided to put our AI to work to discover our own therapeutics and put them in the clinical trials around 2019. And since then, we've managed to nominate 18 preclinical candidates. We now have 7 drugs in human clinical trials. We have 2 in phase 2, so we're progressing. We use AI across every stage of drug discovery and development. So we have a tool called Biology 42. It's a platform which allows you to identify protein targets very efficiently and also kind of rank them by novelty, confidence, druggability, and many other features, even commercial tractability, because as you know, if you're going after a novel target, and if it's too novel, pharmaceutical companies probably would see you in phase 2 complete. They don't like ultra high novelty when you're a little bit later. And you would also be able to evaluate the targets utilizing hallmarks of aging approaches, looking at in which hallmarks each target scores. We have a system that allows you to generate small molecules with the desired properties. So it's called Chemistry 42. So you basically, once you zoom onto your target of interest, instead of looking for a needle in a haystack, you can generate a perfect needle and then synthesize and test. So that ensures that all your molecules are novel, patentable, and have the desired properties. We have a system called Insilico, which predicts the outcomes of clinical trials. And usually we also develop a biomarker for every drug that we take into human clinical trials. Many of those software tools are available. So we actually sell them we license them to a large community. And as the community uses those tools get better and get validated over time. We try to pick the targets that are implicated in aging and disease at the same time. And sometimes it's not trivial because we're going after cancer, but 30% of the papers about the target might be in aging or in associated areas. Our core lead program is in fibrosis. So we decided to go after idiopathic pulmonary fibrosis first, even though that target scored on 6 hallmarks of aging. And surprisingly, I was just at the Longevity Summit Dublin, wonderful event by Obed Gray, which kind of elucidated some new functions of my target. And one of the groups also showed that this target was picked up by Horvath's clock, multi-species clock, as regulated by one of the methylation sites that scores very, very highly. So we're trying to go after targets that are implicated in aging and disease, but we develop them for diseases in a very traditional approach in accordance with all the good drug development practices as a traditional biotech, trying not to cut any corners, nowadays not to make any big statements anymore within realm of credible. But the idea is to try to get the drugs approved for a disease and then use aging clocks to see if they work on aging. Currently, again, in phase 2.
Thanks, Alex. No, I think that's a great point. That's something we all have to figure out, right? How to thread the needle of working on these aging targets with so much promise, but getting them approved for disease first, whether it's dystrophy or IPF or obesity in our case. So let's maybe talk about some practical applications of AI, in biotech, in aging drug discovery. And, with the view too of like many people listening are probably not in biotech, or if they're in aging biology, or even less familiar with AI too, right? So I'd be curious to hear from each of you, like what is sort of the AI application you can't live without? Like, where do you think it's most essential in your pipeline? If you had to pick just one area, where the innovations of the last few years have been really critical. And are there any areas where you maybe thought it was going to be awesome and we still have some work to do? Yeah.
Does drafting HR documents with ChatGPT count?
That sounds much less painful that way. Yeah.
No, it's such a good question. I mean, I see a lot of it is kind of two stages. So one, we use neural networks and AI to actually do data extraction. So that's a lot of our pipeline is it allows us to move very, very quickly and very, very accurately. As we're growing, what we're finding is that now that we've kind of plumbed some of this unseen depths in chemical space, we're actually starting to able to now do more predictive, and you're starting to use generative AI on it to find our next set of candidates and actually go with a more structured hypothesis-driven approach versus the unbiased screening. So we get the benefits of both worlds.
Interesting. Yeah.
Great. Yeah, and I'd say at Juvena, it's really kind of integrating AI into sort of every step of the process, from our ability in silico to rank, order, and predict thousands of proteins that have the potential to have disease-modifying effects for a given tissue target based on targeting mechanisms involved in loss of the health of tissues, known as tissue homeostasis with aging. And so we're using also a variety of sort of neural networks to kind of do some of those predictions. But then as we kind of get a little farther in the process, we're also then integrating AI and some neural network-based techniques into our actual in vitro screening, where we're doing things as simple as improving our quantitative abilities to predict fluorescent markers from bright field images, or to quantitate the behavior of our animal models after treatment with a specific protein therapeutic candidate. But as the field is evolving, we're really excited about some of the generative AI technologies and some of the new techniques that are really coming about specifically for predicting protein structure relationships, as well as novel amino acid sequences that can bind particular receptor targets. And so, we're basically, learning and evolving as the field is, and taking advantage of a lot of the new architectures to leverage using them to actually, train models based on our own internal data to customize them for our approaches. So, I think it all is wonderful, and it's really about kind of also making sure that there is in many ways a human in the loop, so that we sort of understand, noise from real data and signal. And it's a combination of kind of, pattern recognition, but also a little bit of hypothesis-driven work integrated into every step of the process for us.
And Alex, let's double click on generative AI in particular, because I know that you leverage that a lot in your work. And could you maybe also just introduce it at a high level? Because we've talked about it a couple of times, but I'm not sure everyone's aware of what it means.
So sure, our first paper in generative AI for chemistry, specifically generative adversarial networks, was published in 2016. And then we published many papers, 2017, 18, 19, utilizing different generative AI techniques to either generate synthetic biological data or generate small molecules with the desired properties. So think of generative AI as AI imagination. So many other forms of deep neural networks and other machine learning techniques are very good at prediction, generalization, pattern recognition. But if you want to generate novel objects, novel data with the desired properties, you have to go generative. And one of the most popular generative AI techniques are transformer-based large language models that allow you to generate text. So in this case, you are basically trying to predict the next token in a sequence. And those large language models are trained on massive numbers of tokens and parameters. So you basically try to compress your understanding of the text universe in one small box, and then try to query this box for the answer to your questions, trying to generate the pattern it learned in generating the next token. There are many, many different generative AI techniques. One of the most popular ones that we utilize for small molecule generation are generative tensorial reinforcement learning systems, where we have a combination of multiple generative techniques that generate molecules with the desired objective function. But at the same time, you combine them with a reinforcement learning module where you've got multiple predictive models constantly evaluating output of generative systems and either rewarding them or punishing them for generating good or bad molecules or some other forms of data, for example, biological data. Sometimes when you don't need a snappy response, like in the case of ChatGPT, you can train those generative AI models and run them for hours or days, and they become very good. So you achieve molecular level precision, as we've demonstrated with our systems that allow generation with the desired properties of very complex molecular structures, very novel. And actually, to answer your question, previous question, like where AI can be applied, and if I were to pick just one area, which one would I choose? That question was asked to me in 2016 by a very prominent venture capitalist, and I said, look, we actually are trying to combine all of these steps in one, right? So in one workflow. And if we disconnect and just focus on just one thing, it's not going to be as beneficial as trying to look across the board, right? That's what pharma does very often. They disconnect biology, chemistry, clinical trials, biomarker discovery, the payout structures, and even like PPR. Everything is completely disconnected. Generative AI allows you to connect the previously disconnected areas, especially the large language models that now can reason. And what we are very excited about now is the ability to use transformer-based large language models and other generative techniques to reason on the top level and now run many other domain-oriented AIs in very specific sequences with the desired outcomes, trying to steer them towards the desired objective function. And a desired objective function is always, in our case, is pick the right target that is moderately druggable, moderately novel or very novel, for which we can generate the small molecule very quickly with the desired properties that will provide the maximum benefit to the patient, right? So how do we go from 0 to the patient in as short of the time as possible with the highest efficacy? And those problems now can be pieced together by generative systems. And that's the most exciting part for me.
That's really exciting. That's true. Let's switch topics to another exciting area. You guys all work to some extent on automation, right? And there's this really nice, interesting interface with AI where you can have AI in the loop and have more hypothesis-free discovery. So I'd love to talk about, your work there and how it connects to AI, what you're doing now that you couldn't do a few years ago. And Ben, I know this is sort of the basis for your whole platform, if you want to kick us off there.
Yeah, no, that's such a good question. I think that, for us, I see it as a accelerator that gets us moving as fast as possible, that lets us actually kind of disrupt some of the traditional paradigms of having to go with a target-based approach. One example that I was just thinking of as Hanadie and Alex were talking, that kind of, I think, proves the point of as much as we can incorporate it in effectively and accurately, it improves all of our research. We're looking at libraries of drugs that have really never been used at scale before. This is a very specific example, and hopefully some of the scientists in the room appreciate this. It's incredibly hard sometimes to work with novel compounds because you don't know how well they're going to dissolve in solution. And if you're working with a couple 100,000 compounds, that can actually be days, weeks, months of technician time wasted trying to get things to work. So we even use our technicians in lab now use a neural network that predicts solubility in solution, so that we can really quickly triage if those are 10,000 compounds in a library, which are the 9,000 that we should prioritize going as fast as possible with, to actually start exploring this space. So I think the more accessible and hands-on we can make the AI tools we generate, the faster we're all going to go as a field. Does that kind of get at what you're asking, Kristen?
Yeah, definitely. I think that's interesting, right? I mean, it's just sort of boiling the ocean to find novel pathways, which I think is important in our field. There's still, I mean, like that, yeah. We'll get to another question related in a minute,
sure.
right? But yeah.
Yeah, and maybe just to kind of add in terms of sort of like the automation, when it comes to, proteins, it's actually quite hard to really do very rapid, high throughput screening where, just the ability to sort of manufacture and have the physical amount of proteins you need to test is kind of the rate limiting factor. And so one of the ways that Juvena has, obviated that or kind of reduced that necessity has been in some of the innovations we've done on the in silico predictions of proteins, where we've developed a model that we've dubbed PQNet, which uses a boosted gradient decision tree architecture to essentially rank order proteins that are enriched in an in silico library of about 2,000 proteins enriched in stem cell secretomes using a variety of their features from biochemical, evolutionary conservation, biophysical, and transcriptional profiles, understanding how those proteins may act in healthy and diseased tissues to rank, order, and predict which ones are actually worth making and testing. And then when we get to the in vitro screening, we're also doing robotics enabled as what we consider high throughput in, for example, 384 well format, and then doing high content imaging and single cell RNA-seq to then continue to really generate a lot of that multi-omics data that we can leverage to better understand the function of proteins, and also incorporating things like genetic perturbations and CRISPR screening to test more targets more rapidly. So, yeah.
So I'll also add, I guess, if you haven't seen my lab, just go to YouTube and search for 6th generation laboratory robotics by Insilico. So it's I have a facility with 6 interconnected rooms, fully roboticized. So I've got multiple islands in every room. And the original idea for this facility was to generate a lot of machine learnable biological data, providing very high quality data for machine learning, predominantly for target discovery. So we can process cells, organoids, primary tissue in humans and animals to identify a variety of targets using different target discovery techniques in a fully automated manner, where we don't even have human in the loop. Sometimes you need to exclude human in the loop from the loop to debias the system and just get the kind of novelty and risk profile that the researcher is willing to take for a specific target selection philosophy. And the system automatically identifies the targets from the samples that you give it, and also tests a very large compound library on the samples that you provide. However, I actually realized that there are certain limitations to robotics. And I think, Kristen, I'm not sure how much you use robotics for BioAge, but you probably realize that in many cases it's not necessary, right? So once you've got $2 trillion of publicly available data from the NIH and other sources from the entire web, and you've got powerful generative AI technologies plus massive biobanks that are available out there, you realize that there is no shortage of targets. So I've got probably several thousand disease target associations, high value with a lot of experimental evidence from the high throughput lab. But once you are going to bet on every single, on one target, you should be prepared to, go all the way and put, $30 million behind the program. And that is, you you actually are very quickly going to get into the area where you are very resource constrained. So what we realized is that the main idea behind the target discovery lab that we've built is now kind of shifting into indication expansion. So since now we have this Inflation Reduction Act, which kind of requires you to go off patent at some point in time, right? So, and that prevents many of the pharmaceutical companies to go into small molecule space, and most likely it will also touch biologics very soon. And pretty much every drug to make it cheaper. That basically, that act regulates how long your patent is gonna last, right? And if it takes you, 15 years to develop a drug, and the patent lasts for 20 years, it means that you only have 5 years to kind of milk it, right? To return the funds to the investors and ensure that it's profitable for people to invest in biotech. And one of the ways to fight it is to, instead of trying to lobby the regulators to give you more patent protection, is to see where else your drug works. And the fact that if the drug works on aging, it's likely to work on many diseases. The question is which ones, right? And which ones would you want to test it in, and which drugs do you want to combine it with? And that's where the laboratory automation facility comes into play in a very powerful way. So you can very rapidly test your already existing drugs with a variety of target hypotheses and combos in a variety of experimental models, and see and evaluate the probability that this drug is going to work in alternative diseases. And if you see the drug that is likely to work in many diseases, you should prioritize that one, right? And because most likely you're going to get much more substantial returns on that drug, but also you're going to help many more patients.
Yeah, definitely. Let's take one step back to aging biology, right? Which is sort of where we all came from as scientists, and where we're trying to work on targets and advance them. So what can we as a field, what are we learning from aging biology that is useful? And how is what we're doing feeding back into aging biology research? Like there's certainly areas like biomarkers, but anything else that comes to mind? Would love your thoughts on fruitful collaborations, et cetera. Hanadie?
Sure. Everything we do at Juvena is really based on this underlying wanting to really understand and target the biology of aging and mechanisms associated with tissue degeneration to reverse the process and really restore what we call homeostatic signaling. So we're specifically really looking at it from the angle of proteins and secreted proteins, and the way that they really are acting as biochemical signals to induce complex signal transduction cascades that will instruct cells and tissues in our body to do things like activate progenitor cells, to promote tissue differentiation, to activate the immune system, to really react to various types of diseases or infections. And so what's really exciting also about our approach of then marrying that to really understanding how proteins secreted by stem cells, which are such a rich source of regenerative, factors, what we're seeing is that by doing this phenotype first unbiased screening, we're starting to really unravel a lot of novel disease biology and even new kind of underlying pathways and mechanisms associated with the loss of our tissues' health and function with aging. So there's so much, that we're doing kind of, I think, in industry, us and others, that it's really kind of opening doors to ways in which not only are we understanding the way we age better, but we're actually leveraging that to target those mechanisms and really restore tissue health and promote health span.
Yeah, I think one of the things that got me excited about being a researcher in this field is aging is both universal but heterogeneous. So, I was lucky enough, this is a little bit of personal anecdote. My great-grandmother lived to 111. She helped raise me alongside my grandmother. She was the oldest woman in Alaska. And looking at even between those 4 generations of our family, all of us are aging very differently. I used to say I'm an aging biologist. Now I say I'm an aging biologist. And so what I love about what we do is that it is somewhat universally applicable, but our challenge, and I think as entrepreneurs and researchers, is to figure out creative avenues that let our foundational insights into aging biology and health get to patients and to market. And so I think all of us have probably felt constrained by the market into pushing down the traditional pharma pipeline. And I think that there is some aspect that those are the needs of the environment that we're in, and we have to work around those. But I think we also have to push ourselves to be as creative as possible. You know, one example that Ora Biomedical, we just started a partnership with the US Air Force and Space Force because astronauts exhibit progeria-like symptoms. So why not try and take geroprotective interventions and use them prophylactically? It's very early stages. There's a lot of work to be done, but that's, I think, the kind of work that we need to do as a field to get to really progress human health in the way that I think aging interventions can be. And I think one of the things I'm so excited about being on this panel with the three of you and your companies is that we're all doing drug development, which, if we think about the access and equitability of our insights, so much more people can be affected by a drug as opposed to parabiosis treatment or one of the more out there therapies that other people are advocating for. So it's just a really exciting space to be in.
Yeah, definitely.
So yeah, and one area that I'm very excited about in aging biology is our fundamental understanding of biology being improved by the science of aging clocks. So since the publication of the first methylation aging clock in 2013 to 2012, Stephen Horvath and Hanum published their first predictors. This field has advanced dramatically, and we started gaining this ability to track aging in time and to also try to correlate specific types of biological processes with those aging clocks, and also try to link it to phenotype. Generative AI transformed this field beyond recognition. So now we can feed and separately tokenize multiple different omics data types into transformer-based models that learn to generalize between methylation, transcriptomic, and phenotypic data types, learn the interdependencies between the different features in those data types, and enable you to either identify promising protein targets, get new biological insights. Now we can basically chat to those biological data types. We're planning to open source our Precious 3 GPT soon. Have a look at our Precious 1, and Precious 2 is being published right now. So those tools allow you to generalize among the different data types, get new insights. And I actually think that nowadays, generative AI systems, once you want to teach it the concept of time, you have to teach it aging. And that the only way is to teach it and train it on longitudinal data from cradle to the grave, and everything in between, preferably with all the epigenetic and metadata associated with it, so we can see what kind of factors and stress factors affect our aging processes and what happens in between, and correlate that with disease. Now transformer-based models allow you to do that, and I think that this is the area where we should focus more nowadays than anything else. Transformer-based models for multimodal, multi-omics, multi-species data analysis.
Alex, can I ask you? Can I ask a quick question, Kristen, if that's
Sure.
all right?
Yeah, of course,
Alex,
go for it.
I love the fact that you're making some of your tools open source. Do you worry about calling it Precious GPT, given the use case for the original Ring of Power was to undermine and influence the people that it was gifted to bring power back to the Dark Lord? I just, it's a technical question, and I'm sorry, but I had to ask.
Very good question, but I think that they forgot to open source that ring. So instead of that, they decided to melt it, right? If they were
Enough
to,
of them.
turn it into dust and give it to everybody. I'm pretty sure it could have been better, and they could have maybe put it to a good use, right?
I guess it theoretically was a
that I
longevity
have-
intervention.
Yeah, well, it
Sorry.
was actually anti-longevity intervention. If you look at Golem, look at what happened to him, right?
We all hope we can do better than that.
Yeah, I think it's a good principle of extended life at poor quality of life. His health biomarkers were. Sorry, I have fully derailed this, and I really apologize,
Excellent, awesome
Kristen.
question, Ben. On that note, it's about time for us to shift gears and move to the audience questions. Before that, one super quick round. I'd love for each of you to share one technology in the space of AI and drug discovery that you're excited about and what you think it'll mean for the field in 5 to 10 years. Who wants to go first?
I mean, I can kind of start. I think I'm going to sort of chime in and really support a lot of what Alex is saying with a lot of the generative AI techniques. I think what we've become so excited about recently at Juvena is really leveraging some of these transformer models, these protein language models, and open access resources such as AlphaFold in order to really identify and predict new kind of amino acid sequences based on natural protein sequences where it's improved versions of these proteins with greater stability, stronger, binding, greater efficacy. So I really see so much potential for just how this is going to revolutionize the way that we're making new molecules, not just small molecules, but proteins and probably, larger macromolecules going into the future.
Yeah, I'll jump in and say I'm really excited for the advent of more in-depth AI-driven tools to measure human health biomarkers. There's such incredible working out there with all the different clocks that are available, but I think we need more as we think about trying to get our treatments into actual clinical practice. I'd highlight, the work of my former PI, Matt Kaberlein, and the Optispan group that he started. I'm lucky enough to be a trailblazer on and be a beta tester for all of the different diagnostics that they've run. And it's incredible seeing how your aging biomarkers can, even with just lifestyle intervention, much less a drug intervention, shift in just a matter of time.
Well, I am excited about 3 things. So one, of course, generative AI, transformer-based models for multi-omics, multi-species understanding, and at the same time, you can query them for targets, discover drugs, and use the same transformer to then evaluate the efficacy of the intervention. So very excited about that. I'm actually very excited about your company, Kristen. So I like this concept where you can search through massive amounts of longitudinal data nowadays that is becoming more available, also with generative AI, and then in-license and put the drug into human clinical trial faster. I think that this kind of approach is pretty pioneering, and you should talk a little bit more about your company as well here, even if you're moderating, because I think that if I were to do it all over again, I would probably not do my own chemistry, because the probability of success, where we are, I could have failed like many times, and you've seen other companies failing in the same area on chemistry. Very easy, but I think that we need to find new uses for drugs that are currently in development and do indication expansion into age-related diseases.
Yeah, no, thanks, guys. And yeah, totally aligned, Alex, right? It's very much like your strategy of looking at different indications. There are so many other molecules out there that have these broader potentials. All right, we have a lot of audience questions. So we're just going to get started and see what we can get through by the top of the hour. So first one, what do you think the field of aging biology needs to focus on in order to help biotech and companies like yours? I guess from a science and maybe also a talent pipeline kind of question. Yeah.
I mean, for me, I think it's, the issue for a lot of this is kind of that bottleneck of as you kind of go through development. I think on the kind of research side, the innovative side, the platform side, we're all doing fantastically well. But it really comes to that point where now we have to kind of grow up and mature into these drug development companies where more traditional investors that traditionally write much larger checks are needed for us to get our products to market and to help people. And there's just such huge bottlenecks in sort of regulatory and acceptance, I think broadly globally, for this, approach of healthcare versus sick care. Yeah.
Yeah, I'd like to resonate with that. And I think that what the industry needs is one drug approval
Yeah.
for a dual purpose therapeutic that targets aging and disease at the same time. So with GLP-1s, we saw some kind of early birds of kind of that process happening where they targeted a core biological process, got lucky, and now people who are wealthy and obese at the same time can now be wealthy and not obese. So and maybe live a little bit longer. But at the same time, very few people really had hands-on experience in our industry, had hands-on experience with drug development. You go to any drug discovery conference for aging research, for example, and there will be very, very few people in the room who ever nominated a preclinical candidate. There will be even fewer who did a phase 1 study, right? Even fewer who did a phase 2. So sometimes there's like no hands in the room. And I think that one of the reasons for that is those processes, they take a very long time. You really need to stick with a program with one company for about a decade or more and spend your life doing that and not something else. And that requires dedication, and we need to get some approvals and show to the investors that it's commercially viable. If we don't show investors that this is a gold mine, we can, appeal to altruistic reasons and altruistic emotions all we want, but unless we put some real capitalist drive behind aging research, we're not going to see an avalanche of companies popping up and going to the clinic.
Yeah, I'm going to add to that actually and chime in here because I think there's so much good science in aging biology. There's like a lot of ideas that can be translated. And I think we are held back a little bit by that there's, relative to other areas, there's sort of less of an interface with industry, right? Like to your point, Alex, like a lot of conferences on oncology, there's a huge biotech presence, there's a huge pharma presence. That's not true in aging yet, with the one exception of Alex's conferences right there.
Yeah.
But like, like you have a few pharma coming, right, Alex? And like they're willing to talk to us now. They're interested, like Novartis, Amgen, like they're interested in aging biology. They want to hear our ideas.
Yeah.
Eli Lilly, of course, can't forget Lilly.
So your friends as well,
yeah. But I think there's a lot of willingness on their part to sort of engage, right? And there's a lot to be learned on both sides in terms of the science, but also in terms of the challenges of translation.
Yeah, echoing that, I think it's so great that the ARDD is, I look forward to it every year and looking at the talks that come out of it. I think that, one of the things to flip it around a little bit is showing the researchers in academic aging research what it looks like on the other side. And so just like I think you were getting at, I've been spending 10 years going to aging conferences, and maybe there's 1 or 2 companies that show up. And I think that we lose a lot of potential talent because they don't, academia is not for everyone, clearly. And academia has its own strengths, but we have to show these, developing and younger scientists that are in training, what are the potential outcomes? Because then they'll invest in their own training and then funnel into our actual talent pipelines. And I really, I'm excited about that. I love the idea of education and more. And it also improves the likelihood that they as educators and ourselves as community members are telling, traditional VC that aging is, as Alex said, a gold mine. And, we're no longer digging with shovels. We've got backhoes. All 4 of our companies have these great platforms. And, it takes actualization of that.
So we have a number of different questions all around FDA and FDA's engagement with the concept of aging, what that means. So I'm just going to read a couple of them and then we can engage with them. So 1, does the FDA recognize aging as something that can be targeted or treated for clinical trial purposes? If it did, would that impact the pace or impact of your interventions? And if it didn't, would that be a bottleneck to your work?
Actually, I can very quickly answer this question. I think, Kristen, you should be answering this question because you know this kind of, you have a black belt in this. I just wanted to say that I love the FDA. Those 3-letter words I previously kind of really disliked before I got into clinical trials. Now, I think it's a well-oiled machine. that really wants to talk to you. And they have been polished by many patient advocacy groups over the past couple decades to be super responsive, to be pro-patient, to be pro-company. They really want to ensure that it's safe and effective. They won't let bad things pass. But they have very specific response times that they have to adhere to. So that's kind of the intro. You don't need to recognize aging as a disease for getting aging drug approved, because investors, they actually want you to approve the drug for a specific disease. And it might be an easier way to do that because, imagine a phase 3 study for aging. You would probably need to have a massive number of patients, pretty much like for obesity. That's probably not something that you would want to do commercially. You would want to try to get a drug approved for an indication that is age-related, onset 60, 65. So that's why we decided to go for IPF. And then try to, with minimum resources required, and then try to prove that it works somewhere else. If it does, you would be able to do indication expansion if you're fast enough within your patent life. So you really don't need to get aging recognized as a disease. It could actually be an impediment, in my opinion.
Maybe just to kind of, echo Alex there, while the FDA doesn't recognize aging as a disease, there are many diseases of aging, as well as chronic conditions such as obesity that are associated, and due to unhealthy aging. So there are, in fact, a lot of ways that we can get our therapeutics to market by targeting a specific, disease, but then expanding. And so, for example, with Juvena, our first therapeutic that will enter phase 1 this fall activates AKT signaling specifically and really acts to enhance muscle regeneration, muscle metabolism. And we're seeing that it's showing efficacy across not just aged animals, sarcopenic animals, but also a variety of diseases that have this kind of progeroid accelerated aging like associations, such as many dystrophies. So if you're kind of savvy and understand the regulatory process, there's ways in which, we hope to get our products to market first for one disease, but then because it's, actually affecting multiple organ systems or can really target multiple other diseases within that tissue realm, we can expand.
Yeah, I think that, echoing both their points, I think it is, right now we are constrained to going basically after age-associated biomarkers. I do think, though, there is this, to echo again the sentiment of education for people that are not in aging biotech or aging research, I kind of like, again, I came out of Matt Kaberlein's group, so we're all intimately familiar with rapamycin. Basically rapamycin is essentially, where medical marijuana was 20 years ago, where if you have enough money and you can find a doctor,
that was
you can
good
get
one.
it prescribed. No, it's true. And I think that despite FDA not approving aging, there's people out there taking stuff for aging right now. And these are FDA approved molecules. And, I know it's going to be a long barrier to get that to the point of actually really getting something out there for aging specifically, despite the wonderful market cap, if that were to be the case for any of our compounds, right? But I think that in this context, we as longevity biotech do kind of have this obligation to be somewhat creative with how we go after these. And I think everyone here has kind of acknowledged that you're going after aging biomarkers. But, if we think about what's going to move the needle forward, any success on goal, as Alex said earlier, is going to push the needle. And so that may be an off-label interaction from one of the things that we've good at for a disease. It could be even getting it through companion animals. The Dog Aging Project, I know, is ongoing with trying to get things moving in that front. But even that alone would be a huge indication and may have a lower barrier than the traditional clinical trial that we think about for aging, that we probably can't do right now.
Yeah, the dog strategy is super creative. I think for those of us working in humans right now, there's really two main points, right? Like one is that, as we've touched on, these aging drives disease. So often these aging targets, most of the time, these aging targets are also disease relevant, right? Which is sort of scientific / philosophical point. But the other one, which is really important, is that it's just really a practical point, right? Like if we are going to do an aging trial in phase 3, it's going to be huge. It's going to be long. If you have, call it 50, 100 million to deploy on trials, what would you rather do? Make one big bet on one mechanism or make some more smaller bets on sort of more focused disease specific things, right? So it's challenging, even if there were a regulatory path. That in many ways would still not be sort of the right strategy for most of us, especially for new medicines. I think it might be different, arguably, for things like rapamycin or metformin, where there's human data already. But for new medicine, we don't quite know what they're doing yet, or even if they're safe, a different strategy will probably also still, be the right one. Yeah.
Totally. And I really defer to your experience actually being further down the clinical pipeline than we are right now. Do you think that if there was better biomarkers of human health that you could look at over 6 months to a year in terms of looking if something is truly geroprotective, do you think that might make it easier to get some of these to patients sooner?
I think that, biomarkers are certainly useful in lots of clinical indications, right? So in many cases, even though they're not surrogate endpoints yet, in the sense that FDA will use them to approve a drug, we can still look at them, right? And they're still incredibly useful from a research perspective and giving us sort of confidence about these mechanisms. And useful in the practical sense too, of hey, fund my giant trial off-label in this giant population, look, it moves the biomarker, right? So I don't know how much the regulatory aspect of it is that much of a barrier. But yeah, I think there are certainly intermediaries that frustrate me, right? I would love there to be a regulatory path for sarcopenia and frailty, which I think is important. Yeah. And there's sort of milestones along the road, which I think we still need to work on as a field. Yeah. Let's see. All right. We're almost done. So maybe I'll just ask one question again, going back to talent. And as a young scientist in the field, what skills would you recommend we start learning to stay current with the rapidly advancing AI landscape?
Kristen, maybe you would like to start yourself first, because you are hiring a lot of young scientists as well, right? And you were a young scientist yourself. I remember you since you were a master's student, and now you're a CEO of a giant company.
Yeah, no, it's a great question. The landscape has changed so quickly, and there's so much online now too, right? I think you can really immerse yourself in the field, in computational biology in general. You can probably get a start with online courses, but then you can probably also work with a company or get involved and get exposed to what's being translated. The advice I often give personally to people in the field is just go to conferences, Nothing really beats walking around posters and talking to people about their science, learning about what's cutting edge and what's exciting to you, and then learning, once you believe in a problem, kind of learning all the tools you need for that problem, whether it's AI or whether it's molecular biology.
Agreed 100%. I actually think that the best way to prepare yourself for the industry is to try to see yourself as a pharmaceutical industry executive 15 years down the road and plan your career backwards. So go to a good school, don't skip anything, don't cut the corners, try to talk to as many big pharma people as possible. Even Apple learned something from Xerox. And you might want to look at the pharma companies as those Xeroxes, where you can find this mouse that you can repurpose and take it to the next level. So I think that's very important to spend some time in big pharma before you go into biotech. It also will give you some understanding of the regulatory landscape, the traditional processes, and then when you look at the industry from the outside, right, from the outside pharma, it will be easier for you to partner because you will know how those large mechanisms work, and they're not going to go away.
I think, one additional thing to add is just this idea of interdisciplinary kind of learning and training, where, you yourself don't necessarily have to be the expert and, coder that knows how to train these models or build the architectures, but just know what's out there, what's available. And so if you're a biologist and an expert in one area, you can start to leverage the latest, across disciplines and across AI to potentially improve your ability to identify solutions and to problem solve. And so that also entails really partnering with people from other disciplines and kind of just having that open mind.
Yeah, echoing that, I think that's such a great point. Focusing on problem solving and being able to adapt to the new tools. You know, I'll be fully honest. I use ChatGPT to help me write software every single day, but it's about having the higher level idea of how those pieces fit together so that you can adapt to all these tools as they come and figure out the best way to most efficiently use them. It's a great question.
Yeah, it's a good point. Like this whole landscape evolves every year, right? And you have to stay on top of it. And that's also a lot of the fun, right? So, all right, well, let's wrap it up. Thank you so much. Really enjoyed speaking with all of you. I believe there's some slides. Oh, here they come. So AFAR will be sharing and recording on their website, which they'll share over social media as well. And yeah, you can follow AFAR on LinkedIn and Twitter and Facebook. And finally, please support AFAR with a donation to help to continue to build free webinars like this one and circulate them to the community. And yeah, thanks again, all of you. Really enjoyed the conversation.
Thank you, AFAR, and please ARDD. All of us are going to be there.
Yeah, thank you all so much, Kristen. A far out job moderating.
Oh, awesome panelists. So it was easy.
Everyone, and do reach out on LinkedIn and social networks if you have, further questions that we weren't able to answer live. I'm always happy to try to chat with folks, and hopefully if you see me at conferences, come say hi.
Some very quotable moments. Thank you, guys.
Sorry.
Okay.
Take care.
Take care.
Bye-bye.